My name's Cody. This summer I worked with the Shark Population Assessment Group, which is a group of research underneath the NOAA Association. NOAA is the National Oceanic and Atmospheric Administration, which is actually underneath the Department of Commerce, so it is a government organization. And underneath the National Oceanic and Atmospheric Administration, there's the National Marine Fisheries Service. Throughout the United States, there are about 6 different National Marine Fisheries Service labs. I worked at the one in Panama City this summer. Panama City is on the panhandle of Florida over here, and this is the lab that I worked in St. Andrews Bay. And basically this lab is just responsible for research for management plans for marine fisheries throughout the Gulf, Atlantic, and even some of the Caribbean. So within the National Marine Fisheries Service, they have 2 different sections. They have the fisheries research side, and then they also have the Shark Population Assessment Group. The Shark Population Assessment Group is responsible for juvenile population assessments in the Gulf. There are many teams that work together with this association that collect data all throughout the field season, but they also do work with age and growth studies, stock assessments, reproduction patterns. They correlate a fisheries observer program where they get samples from commercial fishermen, and they also do a lot with the sawfish conservation. Working with the SPAG group, they do 2 main field projects throughout the season, and the first one is Gulf span, and the second one is the sawfish conservation. So the first project is the Gulf span, which is Gulf of Mexico State Shark Pupping and Nursery Area. We pretty much just go out throughout the Gulf and sample different nursery areas for juvenile shark species for population assessments to try and figure out where they're pupping, how many, how often, and stuff like that. The data collected is used in the NOAA Sustainable Fisheries Stock Assessment and Fisheries Evaluation Report, which is pretty much where they just decide how they set their regulations on fisheries with recreational fishermen and stuff like that, and commercial fisheries as well. But we would catch blacktip sharks down here. This is a little fine-toothed shark, and then we'd also catch cow-nosed rays and various other elasmobranchs as well. In this project, like I said, we had 4 main sampling sites. We had the St. Andrews Bay, which was in around Panama City and here. Crooked Island Sound, which is actually where this dot is right here. It's just a little east of Panama City. Port St. St. Joseph Bay, a little further east, and then also Apalachicola Bay down in Indian Pass. For these sampling sites, we would use 6 100-foot gill nets, where their size did range from 3 by 3 inches mesh size to 5.5 inches by 5.5 inches. We would let the nets soak for about 30 minutes in between sets, pull them up, and then depending on which species we caught, we would do caudal length, fork length, stretch length, sex, fin clips, take water quality data, and if the animal is still alive, we would tag it and send it on its way. And these are just some more of the species. We have a couple fine tooths right here. And mainly the species that we worked with were the sharp nose, fine tooth, scalloped hammerhead, great hammerheads, and the black tips and spinner sharks. Those are the ones that we saw the most of, but we did catch a pretty big variety of animals. The second project that we worked on was a small tooth sawfish conservation project. This lab is responsible for handling the juvenile sawfish population assessments, and with this job we would only sample in the Everglades National Marine Park. There's two main sections. We would sample in the northern area of the park and the southern area. The park is huge, but when we would sample up here, we would be in the Everglades City at the northern end, and then we would actually stay down here in Key Largo and sample in Florida Bay. A little bit about the sawfish. They are listed under the Endangered Species Act in 1999. And they handle all of these juvenile reproduction studies inside of the park. They do adult studies as well, but the lab that I work for just handled the juveniles. And pretty much with these these animals were endangered, so we had to be especially careful. We couldn't be as free with the nets as we were with the other shark species. We would use 100-foot or 200-foot gill nets with mesh sizes of 3 by 2 inches and 4 by 2 inches. And with these, we would actually just set up against the mangroves or sandy beaches. The smaller baby animals would hang out around in the shallows most of the time just to avoid predation and stuff like that. Data collected for this as well: caudal length, fork length, stretch length. We would get pictures. We would do the rostrum, which is they would do a bunch of studies on rostrum length in comparison to their diet. They would count the teeth. We would identify any kinds of lesions or, spots on the animal that could be fatal. We would tag them, get some fin clips, and release them as well. When we weren't in the field, we worked in the lab. We did wet and dry lab projects throughout the summer. The main project in the lab that we worked on was the age and growth studies. They do a lot of these age and growth studies throughout different species, and pretty much what we would do is the observer program would accumulate tons of vertebrae samples, and they would send them to us, and we would clean them, get them clean. Like up here, some would range from a couple centimeters to a couple inches. We would then section them on a saw, prepare them on a microscope, and then we would read them under a microscope to count how many growth rings they have. We'd also work with fin clips. They do a lot of genetic testing, not necessarily at the lab. They would send them off, but we would do a bunch of stuff with fin clips as well. And this is just an example of the section vertebrae that we would read. As you can see right here, this is actually a sawfish vertebrae section, and each ring, we would just count the rings, and by those, they could tell how old they were. Some other things that we would do, we did do a lot of necropsy and sampling, or taking samples from organisms either brought into us or that we collected. We had about a 10-foot hammerhead wash up on the beach one day that was brought in, collected reproduction tissues and stuff like that for just studies that they had going on down here. Male black-tip shark reproduction, they would collect the claspers and the testes organs as well. And we would also do jaw dissections. This was more for their outreach opportunities, but we would clean and dissect the jaws as well. I'd like to thank the NOAA National Marine Fisheries Lab. Dana Bethay was the research ecologist who was my boss this summer. Also, I'd like to thank Warnell for teaching me about just the fisheries biology in general. This is my last year here in Warnell, and I felt like not only did I take a lot from this internship, but I also contributed a lot by already knowing fish biology, ecology, and field techniques. So, thank you.